important aspects of archaeology is knowing how old something is. One of the ways in which archaeologists do this is through radiocarbon dating. We're here with Carla Haddon at the Center for Applied Isotope Studies, and she's going to walk us through exactly how this process works and what radiocarbon dating is. Oh, Carla, there you are.
Hi, Victor.
How's it going?
Good. How are you?
So, we're here to talk some science and to talk about radiocarbon dating and how old things are. So, first of all, can you explain a little bit to us about what exactly radiocarbon is?
Sure.
How we measure it?
Sure, Victor. So radiocarbon is a radioactive isotope of carbon. Radiocarbon is in every living thing. It's produced in the upper atmosphere, and it works its way through the food webs. First, plants bring in CO2 with this radiocarbon and convert it into biomass through photosynthesis. And then animals eat the plants, higher level animals eat those animals, and so on. So it works its way all through the food web. Once it's in the food web, every living thing has approximately the same amount of radiocarbon in its body. But once you die, that amount of radiocarbon in the tissue starts to decline over time. And so using that, we can estimate the age of something from the amount of radiocarbon in its tissues.
That's fantastic. Now, as I understand it, radiocarbon has a specific decay rate and a specific half-life. Can you explain a little bit about what that is?
That's right. So radiocarbon has a half-life of about 5,700 years. And that means if you start the clock the moment something dies, about 5,700 years later, there's going to be half the amount of radiocarbon from when the clock started. Another 5,700 years later, you're going to have half as much again, and so on. And we call that the half-life. So radiocarbon dating is effective for dating things up to about 10 half-lives. So that's about 60,000 years.
Wow, 60,000
That's right.
years. And have you dated something that's almost 60,000?
We have dated things that are over 60,000 years,
Wow.
but that is the upper limit. All we can say is this thing was at least 60,000 years old.
Okay, so walk me through this a little bit. When someone sends you, an archaeologist sends you a sample, what's the sort of the first steps that you go through here in the pre-treatment lab to prepare that sample for the accelerator?
Okay, well the first thing we do is we try to figure out what the material is made out of. Depending on whether it's wood or charcoal or bone or some other material, we come up with a specific plan on how we want to clean the sample, isolate the carbon within the sample, and then prepare it for turning it into graphite. Graphite is a solid form of carbon that we actually load onto the accelerator. So usually the first step is some kind of physical cleaning. We just remove the outside surface of the sample that's most likely to be contaminated. And then we go through a series of chemical pretreatments using acids and bases to get rid of stuff that's not supposed to be there. As samples are buried underground for hundreds or thousands of years, they take on all kinds of compounds from the soils and from the environment. So we use chemicals to get rid of those compounds and just get the original native carbon signature.
Can you explain a little bit about some of the different types of samples?
Sure.
Bone.
Sure. So one of the most straightforward samples is charcoal. Charcoal is very high concentration of carbon. So all we do in that case is a little physical cleaning and then trying to remove some more big molecules from the charcoal that aren't supposed to be there. Bone is a little more complicated. Bone is a kind of a composite material. We could focus on the protein in the bone called collagen, and collagen is what gives bone kind of its flexibility. Or we could focus on the mineral component of bone called bioapatite, and bioapatite is what gives bones its rigidity. So we can choose to focus on the collagen or the bioapatite, and depending on which process we want to go through, it's going to determine how we're going to handle the samples. So in the case of collagen, we would basically break the sample into very small pieces. We would use an acid called hydrochloric acid to demineralize the bone, so that is getting rid of that hard mineral component. And what we end up with is this gooey, gelatinous substance that we call collagen, and that's what we would use for dating.
Awesome. This leads me to another question. How big does a sample have to be? Does it have to be a giant bone like this for you to date it?
Well, sometimes we get giant bones like that, and we can date giant things like that, but we can also date really tiny things. We can date bones that are about the size of your pinky nail. Other materials have even more carbon than bone does, so we could use even smaller samples. So for example, charcoal and seeds, they have a lot of carbon, so we can date something as tiny as an individual poppy seed.
Fantastic. Thank you so much, Carla. I think we're off to the accelerator now.
Great.
We really appreciate you
Great.
taking the time to explain this to us. So now we've moved from the radiocarbon prep lab to the accelerator room. This is the place where we actually measure how much carbon is left within a given sample. So Carla, what's the next step?
All right. In the last step, we took the solid sample and cleaned it, and we turned it into CO2 gas, and then we turned it back into a solid in the form of graphite. So the next step is to load those little graphite samples onto our accelerator mass spectrometer, or AMS. We have what we call a wheel that we load onto the accelerator. A wheel holds about 133 samples of those little samples of graphite, and we load it onto the ion source, which is here on my right. We load the samples on, and one at a time, the samples are ionized. That means they're given a negative charge. And once they have a charge, we can use magnets to manipulate and move the samples through the system. So we inject this beam of negative carbon ions from the source towards an electromagnet. Once in the electromagnet, the carbon beam is split into 3 separate beams depending on the mass to charge ratio of the carbon ions. What that means is there are 3 different isotopes of carbon in the machine. Carbon-12 weighs the least, so it's going to be controlled or bent most by the magnet, whereas carbon-14 is the heaviest, and it's going to be bent the least. There's another one, carbon-13, that's intermediate in weight. So you have 3 beams, carbon-12, carbon-13, and then carbon-14. That's a way of separating and isolating the carbon-14 from the other isotopes. Once it goes through that magnet, it goes through the center chamber of the accelerator. That's actually the accelerator, where it gives a whole lot of energy to those ions. And it also pushes it through what we call a stripper gas, which reverses the polarity. So before we had negative ions, now we have positive ions that are going to be pulled towards another electromagnet. Once again, it'll go through that electromagnetic field. It'll refine those beams of carbon ions based on their mass to charge ratio. And also at this second magnet, we're going to use what we call Faraday cups to count how much C12 and how much C13 there is in the sample. The carbon-14, which there's very little of, continues on through an electrostatic analyzer here, and then finally it's actually counted at this very small detector at the end here.
Wow, that's fantastic. This is a really large instrument. Why is it so large?
Well, it takes a lot of really powerful magnets to measure these really tiny things. It requires several big pumps and big magnets and that sort of thing to be able to pull it around.
It looks expensive as
well. It is expensive, yeah, a couple $1 million.
Probably shouldn't touch anything in here, should we? So not every place, because it's very big and expensive, has one of these. About how many places around the country can we do this kind of analysis?
There's maybe 10 or 12 labs in the country that have this kind of instrument, and there's maybe 50 around the world.
Okay. And with this particular instrument, we can really measure quite small things, can't we?
That's right, Victor. We can measure things that are as small as 100 micrograms of carbon.
Wow, that's really tiny.
Really tiny.
That's really tiny. So how long does the process for a whole wheel take?
Okay, so we load a single wheel that has about 133 samples, and it takes about 3.5 days from beginning to end to measure the entire wheel. So once we start it, it measures for several days.
Well, Carla, this is fantastic, and now we know how radiocarbon dating works from start to finish.
That's right.
Thank you so much.
You're very welcome.
We really appreciate it.